My name is Elena Piskounova. I'm at Weill Cornell Medicine in New York, and the project title is The Role of NAD Kinase Isoform Switching in Melanoma Metastasis. Specifically, was very interested in the process of metastasis and understanding cancer metastasis at the molecular level. I focused on the metabolic differences metabolic adaptations that occur as cells leave the primary tumor and survive in the bloodstream and colonize distant sites. So part of that work was discovering that oxidative stress limits metastasis. Specifically, I showed that melanoma cells that successfully do colonize distant sites have to overcome oxidative stress through metabolic rewiring. And my lab is very much focused on exploring further the molecular plasticity of metastasizing cancer cells. And what we're hoping to do is find specific adaptations that cells undergo and successful metastasizers require for colonization so that we can identify therapeutic targets. So essentially the idea is that if we can stop them or slow them down from adapting to these new environments as distant sites, they will not be able to take over the organ, which is ultimately what kills the patient. So different cancers have different metastatic organtropism, but melanomas, for example, the type of cancer that we work on, liver is a major site, and some of them actually have ability to go to the brain. Very often, the terminal diagnosis for melanoma patients is brain metastasis because they're not easily operable, but not all melanomas will go there, and that's actually another question where exploring not as deeply, but we would like to, is why are some tumors able to go to the brain and survive and form these metastatic nodules there? And why do some stay in the liver and the lung and sort of more proximal sites? It's interesting because it doesn't seem to be genetic. It's not the mutations. So the changes are not happening in the level of the DNA. But it seems that the cells that do leave the primary site and go on and colonize the organs are very, very plastic. They are very capable of changing their everything, leaving their DNA alone and just changing their transcription and translation, what proteins they produce, what metabolism they rely on, what pathways they rely on. It's some people, and we've talked about this before, it's a little bit like a game of whack-a-mole. The moment you inhibit one thing, the cell changes it something else and is able to survive something else. The survival for melanoma is actually pretty good right now with immunotherapy. Of course, that's been developed much further, but one of the things and the caveats that's coming back, and the reason why we are so interested in understanding the molecular mechanisms within the melanoma cells is that some of the patients who have responded to immunotherapy are now coming back with resistance mechanisms. So it's important to look at both, of course, the immune system, but it's also important to understand what's happening inside the melanoma cell itself and figure out why these melanoma cells are becoming resistant. So one of our goals is to identify these specific hubs, like oxidative stress seems to be a major vulnerability for these cells, and figure out if there's these specific hubs that we can really target just by increasing globally oxidative stress, because then they'll kill metastasizing cancer cells. But unfortunately, because they're so plastic, there isn't a specific thing that we can nail down. So primary tumors at stage 1 and 2 can be removed surgically and they usually haven't spread anywhere. But as soon as the melanoma reaches stage 3, which means it has spread to proximal or to auxiliary lymph nodes, at that point it's a little bit of a gamble, and nobody really knows why some patients at stage 3 will remain in stage 3 and some patients will progress to stage 4, which is distant metastasis in vital organs.